In this online lecture, we will discuss meiosis. This should hopefully be a review for you as you learned about meiosis in your freshman year. It will also represent the completion of our first class lecture in week 1 of genetics. First, let's remember what meiosis is and what its purposes are. Remember that meiosis only occurs in eukaryotic cells. The same is true for mitosis. Organisms utilize meiosis for the purpose of forming haploid gametes that are genetically recombined or altered when compared with the parent. Thus, meiosis is essential in allowing sexual reproduction, which enables both the processes of heredity and also the introduction of genetic variation from one generation to the next. And remember that genetic variation is really important in organisms because it favors it then provides the fodder for natural selection, and that is one of the key processes in evolution. Remember that meiosis always begins with a diploid cell, or 2N, and ends with 4 haploid cells. So in other words, we're going from one cell that is 2N to 4 cells that are each 1N or just N. Okay? And we're introducing genetic variation, so these are genetically different with regards to chromosome content. They're the same fundamental chromosomes, but they've been shuffled up a little bit if you compare them with the parental cell, that original 2N cell. Meiosis is not part of the routine cell cycle. It doesn't occur in somatic cells. It only occurs in the myocytes, which are specialized cells capable of undergoing meiosis. These are key cells for reproduction, and in humans the myocytes are found in the ovaries for women and the testes for men. So like mitosis, meiosis utilizes the stages of prophase, prometaphase, metaphase, anaphase, and telophase. Unlike mitosis, meiosis does all of these things twice. Thus, each meiosis consists of meiosis 1 followed by meiosis 2. Something very special happens during prophase 1, or the first prophase of meiosis. It's something different from what happens in the comparable stage in mitosis. This special event is called crossing over and is shown in the cartoon depicted on this slide. The cartoon begins showing a cell here, okay, with a nucleus that contains 2 pairs of chromosomes. The red and the blue depict homologs for each pair. Note that they are the same length. The term bivalent is used here to refer to the homologs. So blue is from dad, red is from mom. This is a 2 chromosome pair organism. Maybe it's like a spider mite or something like that, okay? The beginning drawing is showing the cell just before it goes into S phase, so DNA replication has not occurred yet at this point. Okay? S phase occurs and it's not shown, and the result is the second step of the cartoon here. And this is showing us that we have now doubled up each DNA molecule or chromosome, using those terms a bit sloppily, but that's okay. Each chromosome's been replicated, and cohesin proteins, COHESIN, I mentioned them in the previous lecture, are helping to hold these replicated chromosomes together at the centromere region, shown in here and in here. Okay? Sister chromatids have effectively been formed. Each sister chromatid has a homolog. Red is one, blue is the other, just like before they were replicated. So these red guys in here are the homolog for these blue guys in here, okay? These are homologs. Together, the entire structure can be called a tetrad. It can also be referred to as a homologous pair, even at this point, okay? What happens next is a really amazing feature of meiosis and a really important aspect of sexual reproduction, and that is that non-homologous sister chromatids pair. This pairing is called synapsing or synapse. Synapse is spelled here. Okay? the strands of DNA will actually break, allowing swapping of short homologous regions or sequences. This can be seen as it happens through formation of structures called chiasma, which are indicated in this third drawing in the figure. The synapsing that is shown and that results in chiasma is facilitated by proteins that play a role in DNA recombination, as shown, and also in DNA repair. An example of a key protein involved in this is the REC-A protein, which we'll discuss again later in the class. So when these non-sister chromatids pair, so we're seeing that here, and we're seeing that here, okay? The chiasma occurs, and what happens are DNA strands break and then reattach, allowing these regions where swapping is occurring. And one of those regions is in there, okay? So they're actually trading sequences. So if you had a gene there, and the maternally derived chromosome, which is kind of orangish red here, had one allele, and the paternally derived chromosome had a different allele, then you would be trading those alleles. And the result at the end of this meiosis was when we actually ultimately get 4 haploid cells is that you're going to have a genetically different long DNA molecule or chromosome. It's really important to remember that it is the non-homologous sister chromatids that pair. If the sister chromatids paired with one another, then when they swapped genetic material out, no difference would be introduced and we would have no genetic variation. So it's important to remember that it is the non-homologous sister chromatids that are going to find each other and swap out. And that process is referred to as crossing over, okay? We call it just generically crossing over, right? So that's a really important event. Again, it happens in prophase 1 of meiosis. And it's one of the 2 major ways whereby meiosis facilitates the introduction of genetic variation. It's so important, in fact, that if crossing over doesn't occur, there are checkpoints built into the meiotic process that will block meiosis from continuing on. Very, very important process. Once crossing over is complete, the cell then continues through the remaining stages of meiosis 1. These are metaphase, anaphase, and telophase, again all within meiosis 1, the first half of meiosis. Note that during metaphase 1, the homologous sister chromatids, which are called tetrads, line up on the metaphase plate. Then during anaphase 1, one homolog from each pair will be pulled to a pole of the cell. So notice that in this phase right here, notice that the sister chromatids are still attached, okay? They're still attached. The cohesin is attached. It is cohesive at this point in time. Following anaphase 1, we have telophase 1, and then we're actually going to have cell division. So we will have 2 cells that come out of meiosis 1. In these 2 cells, if you compare them with the original cell that began meiosis, the DNA content is halved. In other words, the ploidy is halved, or half of what it began. It is N. So even though we've still got, sister chromatid structures, we still consider these to be actually haploid cells. At this point, we're going into prophase 2. And in prophase 2, not very much happens. The cell just sort of continues to move on in meiosis, and we refer to these structures as dyads at this point in time. Sorry, there's a lot of terminology in meiosis. The dyads will assort independently of one another, and they will be moving or be moved towards the metaphase plate, but will not be fully aligned there yet. And at this point in time, meiosis 1 is complete. In meiosis 2, we again have prophase, metaphase, anaphase, and telophase occurring. Metaphase 2 is the stage at which the dyads, or sister chromatid structures, are aligned on the metaphase plate. Note that the order in which the dyads are aligned on the metaphase plate is random. This is very important to remember. It does not have to be the order shown here. It could have aligned, for example, so that the recombined chromatids were on the top in this view, or it could have aligned in other ways. The randomness of this alignment contributes to Mendel's law of independent assortment and to the introduction of genetic difference in gametes relative to parents. I'll touch a little more on that in a moment. Anaphase II is when the sister chromatids actually disjoin, okay, and are pulled towards the poles. Note that the cohesin had to be broken using the enzyme separase to allow them to disjoin, and that they are being pulled using the kinetochore spindle microtubule contraction mechanism that I mentioned in the previous class. In telophase II, the sister chromatids are at the poles and then split into 2 nuclei and 2 cells. We went into this with 1 cell. During meiosis I, we obtained 2 cells with haploid genetic content. The ploidy does not change during meiosis II, it's constant, but we do have another cell division occurring, and we are left with our 4 haploid cells. Note that independent assortment happened at the stages where the sister chromatids disjoined in meiosis II. It also happened earlier on when the dyads, the intact sister chromatids, disjoined in meiosis I. We're left with 4 cells, each has haploid genetic content, and they're genetically reshuffled relative to the parents. Let's consider that genetic variation in a little bit more detail. This cartoon depicts how crossing over during prophase I can introduce genetic variation. On the right, no crossing over is occurring in this organism with 2 chromosome pairs. As a result, we're left with only 2 possible types of gametes. On the left, crossing over occurred, and we obtained 4 genetically different gametes. This means more genetic variation is introduced as a result of crossing over. Now let's think about the genetic variation due to independent assortment during the meiotic process. The randomness of how chromosomes can align on the metaphase plate and the result of that randomness. Again, we're considering our 2 chromosome paired organism. Again, the red is maternally derived, the blue would be paternally derived, as we've been seeing throughout this lecture. In an organism with 2 pairs of chromosomes, there are 2 possibilities for how they can align on the metaphase plate during meiosis, and these are both shown here on the left and the right. The 2 possibilities ultimately result in 4 different types of gametes. So this can be calculated as 2 to the exponent 2, okay, or 2 to the 2, which equals 4. If we had an organism with 3 chromosome pairs, there would be more possibilities for how those 3 pairs could align on the metaphase plate, and you can calculate the number of possibilities and therefore the number of possible gametes using the formula 2 to the 3, which is 2 times 2, which is 4 times 2, which is 8 possibilities. Now let's think about humans. We have 23 pairs of chromosomes, so the possible different types of gametes we can get just due to independent assortment is 2 to the 23. I forget the exact number, but it's greater than 8 million. You can use your calculators and determine that. I can't do that in my head on the fly. So if you think about that, and you think about, just the variation in the humans that you see around you, or even within your own family, this really explains why you're not much like your siblings, and in fact you're pretty different from your parents too, I'll bet. If you add on top of that the genetic variation that can be introduced due to crossing over, you'll realize that it's in fact somewhat hard to grasp or even calculate just how many different kinds of genetic variations one single human can produce. It's really pretty cool, and it's kind of like part of the miracle of sexual reproduction. Okay, so that concludes the lecture content for this first lecture of the semester. Let's talk a little bit about homework next. Using your lecture notes in your textbook, please complete the Mastering Genetics homework that's been assigned. The due date is indicated here. You should also read Chapter 3 for next class and start to take a look at the problems within the chapter. We'll be moving into the application of the knowledge you should have in meiosis, applying that knowledge to problem solving. And I will see you soon.